Good morning, DBM3A. So you should know that you will be having a presentation about your report for microbiology today, right? Are you ready? Okay, let's proceed to the first reporter, Fernando Salunga, for the characterization of bacterial culture in nutrient broth. Please start.
Good afternoon po, Doc. Good afternoon, classmates. So, here's my report on the characteristics of bacterial colony in nutrient broth or liquid culture. 5 characteristics of bacterial colony that may be observed in liquid culture: surface growth, subsurface growth, amount of growth, sediment, and pigmentation. There are 4 types of surface growth: ring, pellicle, flocculent, and membranous. For ring, Flavobacterium columnare. For pellicle, Mycobacterium smegmatis. For flocculent, Pleurotus austriatus. For membranous, Lentinula edodes. 4 types of subsurface growth: turbid, granular, flocculent, and flaky. Staphylococcus epidermidis demonstrates the turbid subsurface growth. Mycobacterium smegmatis demonstrates granular subsurface growth. Flavobacterium columnare demonstrates flocculent subsurface growth, and Bacillus subtilis demonstrates flaky subsurface growth. There are 3 levels of amount of growth: scanty, moderate, and abundant. For scanty, Mycobacterium smegmatis. For moderate, Staphylococcus epidermidis. For abundant, Lactococcus lactis. There are 4 types of sediment in liquid culture: granular, flocculent, flaky, and viscid. For granular, Lactococcus lactis. For flocculent, Streptococcus pneumoniae. For flaky, Flavobacterium columnare. For viscid, Bacillus subtilis. Some bacteria change the pigmentation of the medium. Escherichia coli in brain heart infusion medium with hydrogen tetrachloroaurate changed the color of the medium into purple.
Okay, thank you, Mr. Salunga. Next reporter, Janine Santos, for the characterization of bacterial culture in agar plates. Please start.
Good morning, Doc. Good morning, classmates. I'm Janine M. Santos, and my report is about characterization of bacterial culture in agar plates. And I have here my presentation, so let me share my screen.
Thank you, Miss Janine Santos. Next reporter, Odilio Van Santos, for the characterization of culture in agar slant. Okay, please start.
Good day po, Doc. Good day, classmates. My name is Odilio Van B. Santos, DPM3A. I'll be reporting this day, so ready na po yung report ko.
In this presentation, I will discuss the characterization of bacterial culture in agar slant. Here I have examples of bacteria and they will identify their culture characteristics. But first, what is an agar slant? It simply is a pop-off or a screw cap tube partly filled with an agar mix and letting the medium solidify at a slant angle. They are used for storing pure cultures for a moderately long term. And their advantages, they reduce the risk of contamination, although they are less observable than those in petri dishes. The first example of bacteria is the Bacillus subtilis, found in soil and in gastrointestinal tract of ruminants and humans. Their amount of growth, they are slow growers, they are scanty. Their color of colony, fuzzy white or slightly yellow. Their texture or consistency, they're brittle or dry, friable. Their optical characteristics, they are translucent. And their form of growth, beaded. Their odor is like a strong foot odor. The next example is the Mycobacterium tuberculosis, found in water and soil. Their amount of growth, they're slow growers, but they grow moderately. They come with an off-white color. They are brittle, opaque. Their form of growth is effuse, and their odor, stale beer. They smell like stale beer. Another example of a bacteria is the E. coli, found in water or food, raw veggies, and undercooked ground beef. They grow abundantly since they are fast growers. They come with an off-white to beige color in colony. They are brittle also. And they are opaque and glistening, which means they are glossy. Their form of growth is echinolate, and their odor is like a stench odor. Here in the category consistency, we have a butyrus bacteria. An example of bacteria is a Shigella. A viscid bacteria, an example is a Staphylococcus aureus. Optical characteristics category, there is opaque, translucent, iridescent, fluorescent, glistening. An example of a translucent bacteria, Shigella and Bacillus subtilis. Fluorescent bacteria is Alii vibrio fishery. They're from the deep water organisms. And here's an example of a iridescent bacteria, although there isn't one in an Agar slant. In the category form, there's a filiform growth pattern, a echinolate growth pattern, which is an example is E. coli and Alii vibrio fishery. There's the beaded pattern. An example of this is the Bacillus subtilis, a fuse pattern. An example of this is a Mycobacterium tuberculosis.
Okay, thank you, Mr. Santos. Next meeting, we will further discuss about the characterization of bacterial culture. For now, that's all for today. Before I forget, we will be having a short quiz after the discussion next meeting, so be prepared.